Okay, so welcome back. So we're going to talk here about meninges, ventricles, CSF, and something called the blood-brain barrier. So whenever we look at a section of our scalp, so we look at a section of essentially our skull, all right? So if we look here, here's our skull, right? So the bone, here's hair up here, and here's the brain down here. We have these three layers. We have these three layers called meninges, all right? And these meninges are connective tissue coverings that surround our brain and also surround our spinal cord that provide them that provides the brain and the spinal cord both with protection. All right. So from deep to superficial, we have the pia mater here, we have the arachnoid mater in the middle, and we have the dura mater more superficially. So we'll talk about these in more detail here. So these are connective tissue layers. They serve to surround and support the brain, but they also serve to do the same thing around the spinal cord. We'll see that there's a few differences between those that surround the brain and those that surround the spinal cord. They also protect and enclose blood vessels here. They circulate cerebral spinal fluid that we'll see in this space right down here called the subarachnoid space. And they are largely a protective feature of our brain. So you've likely heard of meningitis. And so if you're, whenever you came to college, whenever you came to university, you had to have the meningitis vaccine, right? And so meningitis is a disease or is a inflammation of one or more of these meningeal layers here. So we'll talk about them from deep to superficial, starting with the pia mater, going to the arachnoid mater, and going to the dura mater. So most deep on the surface of the brain, we have the pia. So the term pia means thin or it means delicate. Okay, so this is the thin, delicate layer, and it is directly on the surface of the brain. So this term right here, it is adhering to the brain. You cannot physically remove the pia from the surface of the brain. All right. So when I did my gross anatomy and we did our cadaver dissection, we took out our cadaver's brain. We examined it. We looked at the dura. We looked at the arachnoid, the pia. The pia is on the surface of the brain. If you were to try to even attempt to remove the pia, you would destroy the brain tissue itself. So that's why this term here is just sitting on top of the brain, because it's representing the fact that the pia is sitting on the surface of the brain as well. Okay. So we also have the arachnoid mater. So you may be thinking that arachnoid, this sounds familiar. You may have heard of arachnophobia. So arachnophobia is a fear of spiders, and so this term arachnoid means spider-like, right? And the reason the early anatomists named this arachnoid mater is because of this space down here. The subarachnoid space contains these things called arachnoid trabeculae that we see right here. These are little extensions of the arachnoid mater, and the anatomists thought that it looked like the web of a spider, so they called this the arachnoid trabeculae. Now, the important thing about the arachnoid trabeculae, it's in the middle, so it's between the pia and the dura, is that it contains this space here. And this space is called the subarachnoid space. All right. This subarachnoid space contains and circulates cerebral spinal fluid around the brain as well as around the spinal cord. And we'll see later how the cerebral spinal fluid is made, but this is an important location. This is also important clinically. If you were to do something called a spinal tap or a lumbar puncture, what this is doing, is what the clinician or the physician is doing, is they're going into this subarachnoid space, specifically in the spine, so associated with the spinal cord, and they are removing CSF. They're aspirating a little bit. They're sending it off to the lab, and they're testing it to see if it contains virus or bacteria to confirm or to rule out a possible diagnosis of meningitis. All right. So our last one is the dura. So the term dura means tough, and for all of these, the term mater means mother. So the dura mater in this case literally translates to tough mother. Okay, and it is one tough mother. It completely surrounds the brain, and it is deep, just deep to the skull. So if you look down here, you can see this layer that I have circled right here. This is the dura surrounding our brain. It is quite thick. It is very distinct. It's very obvious, and it is the outermost. Now, surrounding the brain, it is unique because it comes in two layers, right? There is a meningeal layer, and there is one called a periosteal layer. So what you have are you have areas where it is solid, like this. It is solid, and then it splits into kind of this upper layer, and then it splits kind of into this lower layer, and then ultimately they come back together. All right. And when after they come back together, you see that you've created this space here in the middle, right? So this space is something called a dural venous sinus that we see right here. This dural venous sinus is essentially a blood vessel. It's essentially a vein that you only find with the dura associated with the brain. And what it does is it serves to one, drain blood from the brain, but also serve as a site where we can return our cerebrospinal fluid, or our CSF, back to the blood. So we'll see in just a second that the CSF comes from the blood, and it is ultimately returned back to the blood as well. Okay, so maybe you can see this small opening right here. That little opening is an example of a dural venous sinus, one specifically called the superior sagittal sinus. So we have all of these sinuses in the brain, and these all contain blood, venous blood, and they are serving to drain both blood and recollect cerebrospinal fluid from the subarachnoid space. Okay. You also have an epidural space, which is essentially a potential space, meaning it's not really there. It doesn't really have anything in it, except for if you were to have some type of damage, where blood, some type of trauma to the skull, where you could have blood leak into this epidural space, in which case you could see this on a CT scan. You could see it quite obviously. Okay. So we've mentioned a few times this idea of cerebrospinal fluid, and we mentioned earlier the ventricles, the ventricular system of the brain. So we said that the ventricles are derived essentially from the remnant of that neural tube during development. And what we have we have 4 ventricles. Okay, we have 2 lateral, we have a third, and we have a fourth. So here's our lateral right here, here's our third, and here's our fourth. Within these ventricles, we have cerebrospinal fluid. Okay, and the cerebrospinal fluid is created via this tissue called the choroid plexus. And this choroid plexus is derived from the pia mater. Okay, so there are areas where the pia mater essentially projects and extends down into the lateral and third ventricles. On the outside of the choroid plexus, you have these cells called ependymal cells, which are a type of glial or neuroglial cell. And they're essentially what are responsible for ultimately making our cerebrospinal fluid. Okay, so the ventricles with the choroid plexus makes the CSF. It flows through the ventricles from the lateral to the third to the fourth, and then it goes into the subarachnoid space around the brain and around the spinal cord as well. There's also this small opening called the central canal, which is an opening in the spinal cord, which is essentially a continuation of the fourth ventricle, as we will see in just a brief moment. So whenever we look at our ventricles more closely, we see that there are 4 in total. So if we look at this image up here, we have a lateral right here. We have another lateral. We have this structure right here in the middle, right here that I circled and kind of covered up. That's the third. And then this structure here, that is the fourth. Okay, so lateral ventricles are 1 and 2. Doesn't matter which one you call 1 or 2, just lateral or 1 and 2. You have the third, and then you have the fourth. And again, these are circulating cerebrospinal fluid. So when we look at the lateral, they are really divided and separated by a thin wall called the septum pellucidum. Okay, septum pellucidum. If you've ever done a sheep brain dissection, if you section the brain just right in the middle, you'll find that there is a small piece of tissue. If you section it just right, there'll be this thin membranous connective tissue that is kind of covering one opening of the lateral ventricle, and this is called the septum pellucidum, and it divides the left lateral ventricle from the right lateral ventricle. All right. So from the 2 lateral ventricles, there is a small opening called the interventricular foramen. So what does the term interventricular mean? We're looking at this term here, interventricular. Well, the term inter means between, right? And the term ventricular is just referring to our ventricle. So this is, and if you remember from like skeletal anatomy, foramen is just an opening. So the interventricular foramen means that there is an opening or a hole between the lateral ventricles and the third, and that's exactly what we see. So if we look here, there is this small opening right here, and there is this small opening right here that allows CSF to flow from the lateral ventricles, where it's made by the choroid plexus, into the third, and this is called the interventricular foramen. So here's our third ventricle outlined here. It is in that region called the diencephalon that we mentioned earlier when we were talking about the general brain structure and anatomy. All right. So from the third ventricle, CSF will flow down into the fourth ventricle through something called the cerebral aqueduct. And the cerebral aqueduct is this long, slender passageway going down the midbrain or through the midbrain, and it ends up at this fourth ventricle right here. And then from the fourth ventricle, CSF will go to a couple of places. It can go either into the central canal of the spinal cord that we mentioned earlier, or it can enter into the subarachnoid space that will ultimately surround the brain and then ultimately surround the spinal cord as well. So if we look here, we can see how we actually make our cerebrospinal fluid. So we've mentioned CSF is in the ventricles, it's in the subarachnoid space. Well, the way we make it is through this choroid plexus. So this graph here is a really good representation. So for our choroid plexus, we have three general things. We have these ependymal cells, we have the pia mater that we said earlier, and then we have blood vessels. So the reason we have blood vessels is because our CSF is ultimately from the blood, okay? So it has a similar composition to the blood. Now, it's not exactly the same. There's some differences, but it's largely water is what it is. So what we have are these extensions, and you can see these here in the lateral ventricles of an actual brain sample here. Is this choroid plexus, blood will be filtered out of the capillary. It will cross this wall of the pia mater, and it will enter and go through these ependymal cells here. And the ependymal cells will filter, they will add things, they will remove ions and different molecules within the blood. And then what comes out into this cavity of the ventricle, or the luminal space of the ventricle, is the finished CSF, right? So you may be thinking that, well, if it comes from the blood, does it look like the blood? Well, it does not. So if you think about the color of blood, so blood is red, right? So blood is always red. It's not blue or purple or anything else that you may have been told. It's always red. It can be bright red or dark red. Our CSF is not red. Our CSF is clear, or it's kind of a pale yellow color. So it's more similar to the plasma part of our blood, the liquid component. CSF is 99% water, okay? Blood is red, so CSF is clear yellow. So we know that blood has hemoglobin in it, gives it this kind of red color, it binds oxygen. CSF is yellow, so we know it probably doesn't contain hemoglobin, and it doesn't. And it contains fewer proteins than our blood as well, okay? So whenever we are ready to reabsorb our CSF, or we have made some, it needs to be entered back into the blood, there are these things called arachnoid villi. Sometimes they're called arachnoid granulations. And there are these large projections that you see here of the subarachnoid space into one of those dural venous sinuses that we mentioned previously, right? So we make about 500 milliliters of CSF a day, but at any one time surrounding our subarachnoid space, we have, or within the subarachnoid space, we have about 150. So if we only have 150 at one time and we make 500, this tells you that we have to make it, but then we also have to drain it and get it back into the blood as well, okay? And so the way this happens is through these arachnoid villi, or these arachnoid granulations, that project into the dural venous sinus, so into this blood space here, and ultimately return the CSF back to the blood. So here's a flow, or kind of a outline of how CSF flows throughout the brain. So we know it's made in the choroid plexus that we just mentioned, in the lateral and third ventricles. From there, it goes through the interventricular foramen to the third ventricle, through the cerebral aqueduct to the fourth ventricle. From there, it can go into the central canal of the spinal cord, or into the, into the subarachnoid space. Once we're in the subarachnoid space and we are ready to reabsorb it, we can go into these arachnoid granulations that we see here, return it back to the dural venous sinus, and then now we're back in the blood, and we can ultimately, it's returned to the blood, and we can ultimately make more of it whenever necessary, okay? So these images here I really like. There are three different images, but they're basically showing the same thing. And what they're showing are a couple of things. They're showing the meninges, they're showing the skull, they're showing the brain, but most importantly, they're showing the subarachnoid space, they're showing the arachnoid villi, or arachnoid granulations, and they're showing the dural venous sinus, okay? So this image down here, this is probably the most anatomically correct one, or what it actually looks like. So you have this layer of dura, right? So right here where this red color is, you see that it splits into an upper layer and into a lower layer. And when it does, it creates this opening, right? So we have this nice, large opening, which is our dural venous sinus. Now, within the middle of this, we have this arachnoid granulation, right? That is draining CSF from here. It comes out, and it enters into that dural venous sinus, and it's ultimately returned to the blood. So we see the same thing up here in this upper right image, and we see the same thing here in this upper left image. You have subarachnoid space right here in the left. It will allow CSF to flow up into that arachnoid granulation and be returned back to the blood here, and then we can remake CSF whenever the time is needed. So that this image is showing a video. We're not going to watch the video. Now, there's also something surrounding our brain called the blood-brain barrier. Now, the blood-brain barrier, you may have heard of this. It's mentioned sometimes. And the name of it is a very good description of what it actually does, or what its role is. It is a barrier between the brain tissue, and the blood, supplying the brain with oxygen and glucose and nutrients. So you have these glial cells. So you have this neuroglia, which, remember, neuroglia is a type of nervous system cell, different from our neurons. And what we have are these cells called astrocytes. And these astrocytes have these little feet, these little pedicles, or these little perivascular feet that you see right here that extend around a section of the blood vessel. These astrocytes with their feet surround and kind of seal off the blood vessel and prevent just everything in the blood vessel from just leaking out and coming out into the brain tissue. So all out here is where you'd have brain tissue, right? So these feet are very good at preventing those things from just, or preventing everything from just indiscriminately just leaking out, okay? Now, you may think that, well, we still need certain things to leak out, right? We still want oxygen. We need carbon dioxide to be able to come in. We need glucose. We need lipids, all these other things. And so the blood-brain barrier is permeable to those things, but it is not permeable to other things, all right? So it prevents things like certain drugs, other waste products, abnormal solutes from entering our brain tissue from our blood. Now, what it also does is that at the same time, at certain points, it can prevent hopeful things from passing through as well. If you think of things like chemotherapeutic drugs, right? So this is one of the reasons that brain tumors, brain cancers, brain infections are so dangerous and so hard to treat. It's because the medication has to be able to cross the blood-brain barrier. So normally the blood-brain barrier is a very normal, healthy, physiological structure. But whenever we're trying to treat a brain infection, but the medication can't cross the blood-brain barrier, we're not really doing any good, right? And so it's normally healthy, it's normally good, but it is also, it can be a barrier to work around in a clinical setting, right? So we also have some things that can pass, and these are things like alcohol, right? So alcohol can cross the blood-brain barrier, it can enter the brain tissue, and it can paralyze what's known as the prefrontal cortex of our frontal lobe. Well, the prefrontal cortex controls things like thinking and critical thinking and reasoning and judgment and our motor activity and our personality, right? So this is why increased alcohol consumption, right, can influence all these things, right? So if you increase the alcohol consumption, you have a change in your personality. You have a change in your critical thinking, in your reasoning, in your decision making, right? And it's because alcohol is permeable to the blood-brain barrier. You may have also taken medications like NyQuil or Benadryl or something like this. When you take it after a certain point, you become drowsy, you become sleepy, your CNS is depressed. This is because this medication is capable of being in the blood, getting to the blood vessels around the brain, and flowing out. And when it does so, it enters into the brain tissue. Now, this also brings up another point that kind of has more of a clinical relevance, and this is something called the Monroe-Kelly hypothesis, or the Kelly-Monroe hypothesis. It's sometimes referred to as both. But what this is stating is that there are three things in the skull, so within the cranial cavity, and an increase in one of these has to result in a decrease in the other. Okay. So what these are is we have brain tissue is one, We have blood and blood vessels as the second, and we have cerebrospinal fluid. Those are the three main components that are taking up most of the volume inside our cranial cavity. And so what this hypothesis is saying, this Kelly-Monroe or Monroe-Kelly hypothesis is saying that an increase in one of those results in a decrease, a compensatory decrease in one or the remaining two, right? So imagine you have a blockage in the CSF drainage. So, right, we're making the CSF 500 mils a day. We normally drain it through the arachnoid granulation into the dural venous sinus. Let's say there's a blockage there. And so we can't drain it, but we continue to make it. So we have this buildup. This is going to put pressure on the brain and will cause the brain to kind of become compressed, right? So there's only so much space in our cranial cavity. So there's only so much room for brain tissue, for blood, and for CSF. Well, this blockage of CSF, this is what we see in things like hydrocephalus, right? So hydrocephalus can result in a blockage of the drainage of CSF that results in, or can result in permanent brain damage, and it can also be fatal if extensive enough, right? So this is very, very serious. You can also think of things like a hemorrhagic stroke, where blood is bleeding into the brain. You have this accumulation of this blood. This can put pressure on the brain, and so you can have what's called increased intracranial pressure within the skull, which is not normal and which is not physiological, and which has to be taken very, very seriously. Okay, so sometimes in a clinical setting there can be something known as a craniotomy, in which a region of the skull, region of the cranium, is removed, and by removing it, it relieves the pressure on the brain and allows it to kind of expand, and it decreases that intracranial pressure back to a more normal level. Okay, so we will stop there, and we will pick up talking about cell physiology and how neurons are connected and how they form different types of circuits.